Welcome, and in this video I'm going to talk about the second way we can prepare ethanol, which is fermentation of glucose. Fermentation of glucose. This is also another technique apart from hydration of ethene that we can use to prepare ethanol. And through fermentation, the main thing that you need to know are the sources of this glucose. Majorly in nature, we can get glucose through sugar or even starch. And the first key thing we do, we dissolve sugar or starch in water. It is dissolved in water. And then we add yeast to the sugar or starch solution. Then the system is closed tight. Is closed tightly, meaning we need it to react in the absence of air. In the absence of air, and specifically oxygen. So this shows that we need anaerobic respiration to occur. Anaerobic respiration to occur. We need an aerobic respiration to occur in that given system. So there are key things that as a student, key features of this reaction that we need to know. Features of this reaction. Features of fermentation as a way to prepare ethanol. The key things that as a student or an examiner, you really need to know. And the first key thing that you really need to know is what is the role of yeast in that given system. Yeast here act as a biological catalyst. It acts as a biological catalyst, and through it, yeast is capable of converting sugar into into ethanol and carbon dioxide. Yeast is capable of converting sugar or starch into ethanol. So it is our yeast here that is going to convert the sugar or starch, for those who will be using starch, into ethanol that we need, plus carbon dioxide. Carbon dioxide. Now, because we are working with yeast, and yeast is more of like living cells. So, apart from understanding yeast as a biological catalyst, what are the key features or conditions under which this reaction can really occur? We have to maintain the temperature upon which the reaction is occurring at about 25 to 30 degrees Celsius. So somebody might be asking him or herself, why do we have to do this? The first key thing, yeast cells are living. If the temperature is too low, the reaction rate will be too slow. If the temperature is too high, yeast cells will be denatured, and therefore reaction will not take place. Reaction won't occur. So that's why we have to ensure that the reaction system is maintained at a temperature of between 25 to 30 degrees. Now, the same system, or another feature of fermentation of glucose in the preparation of ethanol, is that we need an aerobic respiration. Aerobic respiration to occur, or a condition that is airtight. We need an airtight condition, or we don't need oxygen in the system. Why? Because in the presence of air, if oxygen is present, yeast will break down glucose into CO2 and water, which is carbon dioxide, and water. Meaning at the end of the day, we will not have our ethanol that we need here. So if the conditions are met, meaning an airtight condition, a temperature maintained at about 25 to 30 degrees Celsius, glucose, which is chemically written as C6H12O6, will yield ethanol, will give us C2H5OH plus CO2. This reaction can be balanced in a very simple way by just putting a 2 here and a 2 here. So when you balance it that way, you'll have to ensure that you put the conditions upon which the reaction is occurring. A temperature of about 30 degrees Celsius. We're using yeast enzymes in an aerobic condition for that given reaction to occur. So if the system is set that way, we will have our ethanol at the end of the day, and this is just carbon dioxide. We'll get it from glucose with the help of our yeast. So since ethanol can be made through fermentation, how do we make ethanol made this way, pure? Because you'll realize that it is very difficult to keep this system airtight, but sometimes air might escape into the system. So to some extent, some water might also be produced during this process. So to make our ethanol pure, we use fractional distillation. Fractional distillation, a technique that separates substances based on their boiling point. Based on their boiling point. Now here, you'll have to know that ethanol has a boiling point of 78.8 degrees Celsius. Water, which is majorly impurity during this reaction, has a boiling point when pure of about 100 degrees Celsius. So meaning these two substances have close but different boiling points. So we can subject them to fractional distillation, in which when the temperature reaches about 80 degrees Celsius, we expect all ethanol to be collected first as the first distillate. Then, as the temperature rises again to about 100, the next distillate that is going to be collected is going to be purely water as the second distillate. Then through that, we can make our ethanol pure or more concentrated, and hence can be used in other areas in life. Now, there are key things that you need to know. How do we remove yeast cells? Remember, we were using yeast as our catalyst in this reaction. We're using yeast. So how do we remove them? So most of the time, as the concentration of ethanol increases in the system, to more than 20%, yeast cells always die. Yeast cells always die. So this is an explanation why whenever drinks containing ethanol are made, for example, wine, whiskeys, gin, call them, those alcoholic drinks, they always contain a lot of ethanol. So these substances are majorly left in stores for longer duration. The assumption is immediately they are prepared, the alcohol content is low. But as they stay longer in the store, the alcohol content increases. Alcohol content increases. So for that matter, we say in the store. That's why you can sometimes hear that a wine has been in the store for 20 years, for 30 years. So the longer it stays in the store, the more it continues, the reaction continues to take place, because the inside the bottle, it is airtight and we have some yeast cell. So the reaction is still taking place and it becomes more concentrated and more concentrated. So as the alcohol content increases within that given system, yeast cells always die. That's the reason why we are not affected by yeast cells. Now, a situation might come. What if a bottle of wine is left open for a longer time? When left open for a longer time, oxygen in air will oxidize alcohol to form carboxylic acid. And this carboxylic acid is what is responsible for the sour taste. For the sour taste, when alcohol is left in air for longer time, for longer duration. So that's why most of alcoholic bottles are always airtight, because when exposed to air, the alcohols in them will react with oxygen in the air to form carboxylic acid, which makes them lose their alcohol taste, and they'll obviously have a sour taste. Thank you very much. That's what I had. Have a wonderful time.